And this is a picture from your book, 4.8, of membrane transporter. And you will see what's happening here is there's a binding site. This is just they're not green, folks. And the solute aren't little purple balls. It's just diagrammatic. So this is just a diagram. It's really hard to draw the protein exactly as it looks. It's much more complicated than this. So what you see is this is the outside of the cell, the inside of the cell, and to get through the bilayer, the solute then binds to a specific binding site. The very act of binding that site by the solute changes the shape of this protein, and it then opens to the inside of the cell, and the very process of being opened to the inside of the cell changes the shape of the binding site so that the solute can be released into the cell. Yes, sir?
Does it have subunits like the channels?
Some of these do have subunits. But what are you specifically asking?
Does it have, like the channels have subunits to the
I see what. No, channels are not usually associated with mediated transporters. Channels are associated with some kinds of receptors, but definitely not these guys. Okay, so where was I going to go with that? Okay, so these are mediators for mediated transport. This is a kind of a generic picture. There are different kinds of mediators. So just recapping what we talked about yesterday, we talked about plain old diffusion from high concentration to low concentration. This is diffusion of the solute through the membrane. The solute either going through a channel or just through the bilayer. And this is facilitated diffusion. And in your text, the little ball on the membrane simply means that some kind of transporter is involved. And again, we're going from high concentration to low concentration, which is why they call it facilitated diffusion. But it really isn't diffusion. It's really mediated transport, passive mediated transport. But that's the name that everybody uses, so that's the name that we will use also. So this is just plain old diffusion right through the membrane. This is facilitated diffusion through a mediating transporter. And then this one is active transport. What makes this active transport? You tell me. We use energy, yes. You have to use energy to go against the current. What else? Yes, it's uphill, right. You're going, when we say uphill, we mean going from low concentration to high concentration. Because in nature, we tend to go from high to low, the top of the hill to the bottom of the hill. But in this case, we're going from low to high. So this is one clue you've got active transport when you're going from low concentration to high concentration. This is figure 410 from your text. Yes?
Do you ever have facilitated diffusion that uses energy?
No, because when you have a transporter that uses energy, it's called active transport.
So that's always against the concentration?
Yeah. Your cell would not want to use energy to go from high to low, because the thing that drives the movement from this side to this side is simply the concentration gradient. Because it's free, the cell is not going to spend energy in order to get it to happen. Does that make sense?
And I mean, it could diffuse on its own, right? But the diffusion just speed up the process?
Well, it depends on the substance. If the solute is too big to go through a channel, or too big to go through the bilayer, then it might have a specific transporter that just transports that one solute item. I'll give you specific examples of these as time goes on. Well, here's one example. This is the diffusion of glucose. This is figure 4.2. Glucose can get through the cell by plain old ordinary diffusion. However, it's not the only way for glucose to get in. Glucose can get into the cell other ways, which we will discuss later. The membrane is partially permeable to glucose. The length of the arrows in this picture represent the magnitude of the flux. So you see, as time goes by and there's more glucose inside, you see the arrow going to the right is getting shorter until we reach equilibrium. So time C here is equilibrium. The green line represents the amount of glucose, the concentration of glucose in compartment 1, and the purple line in compartment 2. One thing I want to tell you, though, this is a little deceiving, because glucose is catabolized on a very regular basis by glucose 6-phosphate. It's almost immediately catabolized once it gets into the cell. So the concentration inside a cell is most usually less than whatever is outside. So you usually have a relatively low concentration of sugar in the cell just because as soon as it comes in, it gets metabolized. I just wanted to make sure you understood that. This kind of diffusion of glucose is an example of facilitated diffusion from high to low, from higher concentration to lower concentration. So you're not likely to end up with this kind of equilibrium with glucose because it's going to be converted almost as soon as it gets inside the cell. This is a different kind of mediated transport. This is primary active transport. Active because it's going to use ATP. That's the bottom line. We're going from low concentration to high concentration, and we're using ATP. In this case, the transporter acts as an ATPase. Now, when I have ASE at the end of a word, what does that mean? Enzyme. So ATPase is able to break down ATP and release the ADP to go back and get phosphorylated again, and then the phosphate group, the inorganic phosphate, binds to the transporter. When the inorganic phosphate binds to the transporter, it changes the shape of the transporter. So there are actually two things that are going on here. You've got the solute binding to the specific binding site, and you've got phosphorylation of the receptor, leading to a conformational change so that the substance then can be transported across the membrane. And I'm not sure if they're trying to show anything with the number of little purple balls on this picture, but the fact of the matter is the concentration of the substance is higher on this side of the membrane than on this membrane. So we're pumping that solute through the membrane into a compartment that has a higher concentration. So primary active transport is like a pump. It acts as an ATPase. It uses the energy of ATP and the excited phosphate group to change the conformation of the protein. And you notice that once it changes its conformation, the binding site no longer is as high affinity for the solute, so the solute is allowed to be released. This is primary active transport because the transporter doing the transporting actually gets phosphorylated. Not to be confused with secondary active transport, which we'll discuss in a minute. But first, I just want to show you an example of an ATPase primary active transporter. This is a very common transporter. You'll find it just about everywhere. We will talk about this transporter a lot. This is the sodium-potassium antiport, and it's called antiport because it pumps sodium out of the cell and potassium into the cell to maintain gradients. I heard someone say the absence of gradients is death, and that's actually true, because if you look at the relationship between compartments in and out of a cell, you'll find that there are no gradients anymore because these pumps are not able to maintain the gradients because there's no available ATP. So ATP phosphorylates, and the solute sodium binds to the specific binding sites. It changes the conformation of the protein. The protein then releases the sodium inside the cell. Outside the cell. The potassium outside the cell then binds to its specific sites, and the transporter releases the phosphate. It becomes attached to ATP again before phosphorylation, and the potassiums then are released inside the cell. Yes, sir?
So phosphorylation, no, it's the first step.
Phosphorylation is one of the first steps. Yeah. Also binding of the specific.
That just happens.
It's just happening because there's plenty of sodium inside the cell that needs to get outside, because we'll talk about why in a minute. Well, maybe not today, maybe tomorrow. But there are plenty of sodiums and hopefully lots of ATPs. So when it's ready, it gets bound to all of those things. Okay, so this is 411. This is a good one to know about. The sodium potassium ATPase is also called the sodium potassium pump. It's also called the sodium potassium antiport, because sodium is going in one direction, potassium is going in another direction. Okay, this is a different kind of active transport. This is called secondary active transport. So in this example, we have a higher concentration of sodium outside than inside, and that's pretty much the usual case throughout the body. You will have more sodium outside the cells than inside. But in this example, we also have a solute that needs to go against its gradient. It needs to be pushed uphill, basically. And instead of using ATP to phosphorylate the transporter has two specific binding sites, and the gradient for sodium drives the transport of the solute into the cell, in this case. So what's happening is the solute is hitchhiking on the sodium transporter. It's similar to primary, secondary is, but the ATP is not being used right here at the transporter. The ATP is used elsewhere on the membrane, on some other protein, in order to pump the sodium out of the cell to maintain the sodium gradient. And it's the sodium gradient that powers the movement of the solute up its gradient from low to high. Yes?
Is it like the solute sneaks in when the sodium opens it?
I guess you could kind of think that way, except it's very official. It's not really sneaking. It's not like this guy opens the door in the back of the theater and this one just sort of sneaks in. It's actually official, okay, because the transporter is designed for this to happen. It's more like a tandem bicycle, and the stoker in the back just sort of coasts while the guy in the front pedals. Okay, that's kind of what's happening. But the guy in the front is pedaling not from ATP, but from the sodium gradient. Was that a confusing analogy? I don't know if it worked. Let's keep talking about this, and it might become more apparent. Here's an example. Active transport of sodium across an epithelial cell. So we have a pump here. It's an ATPase, and it's pumping sodium out of the cell on the serosal side near the blood vessels. Active transport. The process lowers the concentration of the sodium inside the cell. See, the concentration of sodium is higher in the lumen than it is inside the cell, so we've got a natural gradient, and in this case we just have a channel, just a tunnel, and the sodium, it just goes into the cell. and as fast as it goes in, it's pumped out. So this is the way we move something, a polar ion, well, an ion, like sodium, from one side to the other. We have the pump on one side because it's higher over here. You can see the concentration in the blood is higher than in aluminum. But we're going from low to high with ATP, and that then lowers the concentration inside the cell, keeps it low so that we can continue to diffuse. Oh, that one was 422 in your book. So what's happening with secondary transport, secondary active transport, is you've got a transporter that actually is going from high to low, carrying something from high to low, usually sodium, and the solute then just sort of hitches a ride. I think a better analogy to sneaking in might be, I don't know if this works. No, it doesn't. I was thinking of a kid on a skateboard and his friend just hopping on and riding. That doesn't work. Oh well, anyway. This is co-transport of the substance in the same direction as the sodium is going. The sodium is going down its gradient, and the solute is going up its gradient. Yes?
You need the solute to be able to allow the whole process to happen, right?
Right. They both have to bind to the two sites on the transporter.
I was thinking of it like a translator. Like if the person that didn't speak one language was trying to talk to another person, you have to get through.
So sort of this guy is the
It
translator.
has to be there for the communication to happen.
Either that or this is the person with the card key and the badge, and this is his colleague who doesn't have a card key and a badge, and they both go through the door. It's, oh, Costco. I've done that before. When I'm not a member of Costco and I have a friend with a card, and we go in together and we shop together. Okay, so it's kind of like that. Except that sounds more like your analogy of sneaking, doesn't it? Sorry. No, it's more official than that, yes.
Does secondary active transport, does it always use primary active transport in order to have high Na in the extracellular fluid?
The reason it's called secondary active transport is because ATP is used somewhere to maintain the gradient.
So they always use primary active.
So somewhere there has to be a primary active transporter pumping the ion in question, usually sodium, just to maintain that gradient. Yeah, good question. Did you have a question? No, I thought it's a handout. Sometimes you will find a situation where you have the transport going in opposite directions. So in this case, we'll be bringing the solute in the opposite direction that the sodium is going. It's just a different transporter. These are all different proteins with different functions, but they have similar mechanisms. This is also called a syn port. This kind of transporter. And the syn port simply means that the two substances are going in the same direction. This is called, whoops, pen running out, anti-port. So remember I told you that the sodium-potassium pump is sometimes also called the sodium-potassium antiport. And that's because the sodium goes one direction and the potassium goes the opposite direction. That's not what's happening here. Antiport simply means that the two things being transported are going in opposite directions. An example of a secondary active transporter that does co-transport is the sodium-glucose syn port in some cells. Glucose transport is sometimes done by facilitated diffusion and sometimes done by insulin-gated syn port in cells. So the syn port, actually the syn port is not, well, yes it is. So the syn port then will carry the sodium in the same direction as the glucose into the cell because the sodium concentration is less in the cell. Do I need to talk any more about this or shall I go on? Any more questions? Yes.
Is there a kind of active transporter that does not move the solvents, moving certain solvents?
down in the uphill direction, but accelerates the downhill transportation.
What an interesting idea. I don't off the top of my head remember one. Does anybody? Do you know one? Okay. I don't recall ever encountering one, but that's an interesting idea. My thought is that usually the cell will not spend ATP if it doesn't have to. So it'll take the easy way. However, what you're talking about is something to speed up a process. I haven't heard of anything like that. I would think it would just be a bunch of channels opening in the membrane, because you don't need to expend ATP to get things to go down the gradient. What you could do, though, is have more sodium channels, for instance. You want sodium to move, or you have more whatever, glucose, there are no glucose channels. So you have some other kind of channel that is closed and can be opened in response to a signal, a ligand or some other way, and that may speed the rush of the material through the membrane from high to low. You don't need ATP for that. So I've never heard of a mediated transporter that uses ATP to go from high to low, but that's an interesting thought. You know, I like the fact that you're thinking about this. That's a really good way to do this. You all should be thinking about this. That Berkeley paradigm where you take notes or record a lecture and then throw it in the trunk until right before the midterm, and then stay up all night and study for the midterm. Cram, 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 cram. You come into the I've had people come in, I have the test, say, give it to me before I forget everything. And so you give out the test, and they spew, 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 and then 10 minutes later they've forgotten everything. It's not a good way to learn. And I know you guys think it's about getting units, and it's about getting grade points and all that, but Michael, you're studying for the MCAT. Is this useful for the MCAT?
Absolutely.
Is genetics interest? I mean, any of your biology classes.
All biology is very, very helpful.
So you're better off if you learn it well the first time. And the way to do it is thinking about what you're hearing. So blessings on you. And also thank you for asking the question. When you ask a question in class, then other people will be saying, you know, I was wondering that, and I'm sure glad that person asked. So thank you. Yes, is that a question?
Well, I just had a question. It's kind of just particular though. I was wondering, is it SIN porter or with an N?
No, it's with an N. Sin port. Oh, actually, you know what? That's a different thing. Spell checkers! Good girl, you're a spell checker. Okay. So that's secondary active transport. This is just an example of different ways for calcium and stuff to get in and out of the cell. This organelle in the cell could be like the endoplasmic reticulum, for instance. And you see we have an active transporter going from lower concentration to higher concentration of calcium, pumping that into the organelle, and then we have a channel which allows the calcium to flow from high to low. This channel is not necessarily always open. So the channel may be opened in response to some kind of stimulus. And we'll talk about some of those stimuli in a bit. Here again, we have calcium being pumped out of the cell to maintain the lower concentration inside the cell. And then we have a calcium leak channel that allows diffusion back into the cytoplasm. And then, oh, look here in the lower right-hand corner, we have secondary active transport of calcium using the sodium gradient. So instead of using ATP here to move the calcium out, we're using the sodium gradient. Somewhere else, though, on this membrane, what do you bet we've got? A sodium pump, right. Sodium pump or sodium potassium pump, pumping sodium out to maintain the low concentration of sodium in the cell. It's not shown in this diagram. Oh, this diagram is from the 2001 edition of Vander. They don't have it in there anymore. Too bad. I like it. Just because I think it makes it easier to see. Okay. Summary. what we've been talking about, mediated transport. Just wanted to remind you, this is a mediated transporter. This is 4.8 again. Shows you the carriers that have a specific binding site for the solute. The binding of the solute changes the conformation, allowing the solute to go in. In the case of secondary active transport, there's another binding site for sodium or some other substance that has a higher concentration outside than in. Or, in the case of primary active transport, what do we have on here? On this transporter, if this were primary. And ATPase. Is all mediated transport active? No. This could be going from high to low, in which case we call it facilitated diffusion. You need to know the distinction between those, because how can we have a conversation if every time I say facilitated diffusion, your brain says, "Was that?" You really need to know what that is, not because I'm going to ask for a definition, but because I really want you to understand what we're talking about. So I'm going to list for you types of mediated transport. This pen is toast. That's better. That's T. Okay, the first kind is facilitated. Spell checkers, keep an eye on me. And that goes from high concentration to low concentration. And then we have, so this is not active transport. This is passive, like plain old-fashioned diffusion. Then we have mediated transports that are active. We have primary active, and that goes from a lower concentration to a higher concentration, and uses ATP on the transport protein, the mediator. So the transport protein that is moving the solute from low to high actually is the place where the ATP is used, And secondary active, you're still going from low to high, but it uses ATP somewhere other than the transporter. So is that making sense? Any questions about that? Before we finish talking about active transport, I wanted to talk a little bit about one other kind of active transport. And I just wanted to say that often channels are faster than carriers, just because if you have a turnstile on a gate, it's going to take longer to get through, get a bunch of people through, than if you just have an open gate. And that's what the channel is like, is an open gate. Oh, I guess I should show you this. This is no longer in the book. You don't have to know this. I'm just showing it to you because it's interesting, and that is diffusion and facilitated diffusion have a similar curve getting to equilibrium. Whereas with active transport, you end up with the concentration on one side of the membrane, inside the membrane, greater than the concentration outside, so you're actually going uphill. In the case of flux, the rate of flux in diffusion, because it's just right through the membrane, either through the bilayer or through channels, is direct, is very very straight line. The flux curve for facilitated diffusion is more like that of other mediated transports, which are all active, and that's simply because it has to go through a protein that changes shape. That is just to show you how they're similar and not. That has its own curve. It really should have its own curve, because once that glucose gets inside the cell, almost immediately it's going to get metabolized. But that's a whole different, that's a much more complicated story. Okay, so the other kind of active transport is the process of membrane endocytosis and exocytosis. If you had general biology in junior high, you've heard about this one. So we're not going to spend a whole lot of time on it. Gesundheit. When you have endocytosis, materials are brought in. If it's fluid, it's called pinocytosis. And if it's solid, it's called phagocytosis. If you have material being moved out, it's called exocytosis. Not everybody agrees on what constitutes exocytosis. I have a colleague, esteemed colleague, who says that exocytosis is only the release of materials into the extracellular fluid. But I know other colleagues who say no, exocytosis includes the act of adding membrane to the plasma membrane of the cell through this process, or putting transporters into the membrane. So let's say we have a vesicle that has receptors. These are receptors, believe it or not. And what happens is you have fusion of this receptor-laden vesicle. And what happens is when the fusion is complete, you will have membranes that contain these receptors. So some people consider that exocytosis too. I actually do too. But it's not a big deal. I'm not going to ask you to make that distinction, because even the folks upstairs don't agree on what's going on. But the key is endo, meaning coming in, and exo, leaving. And this is usually for molecules that are proteins or aqueous. You're not going to have steroids and lipids going through this process. Why not? Tell me. That's a good question. Why or why not? Say again? They can slip right on through the bilayer. Good. Yes, they don't need to. Is there a question up there? Okay. But there's a special kind of transport called epithelial transport. And that simply means materials are going through an epithelial layer, a layer of tissue made up of epithelial cells. The material must pass through the plasma membranes and the cytoplasm of the cell, and that's how the epithelial layer regulates what gets through. We've already talked about tight junctions. Water can go through tight junctions, not a whole lot, but most other things will not go through tight junctions. So what you have to have is regulation on the serosal side and the luminal side. In this case, channels and pores that allow material to pass through, and transporters. So epithelial transport it's important in a lot of different organs that we are going to talk about. For instance, the kidneys. You have to have regulated epithelium. And you'll see this kind of a diagram a lot as we go through this book. Oh, and something else I wanted to mention. I know you know this, but in order for the sodium or the water to passively pass from the luminal side through the cell onto the serosal side, is for there to be a gradient. And guess where the gradient comes from? Tell me, where's the gradient coming from? What is the solute making the water concentration less over here? Sodium. So we've got a sodium gradient. And the sort of rule of thumb is water follows the sodium. So if sodium is being pumped across a cell, you can pretty much bet a water will be drawn through the cell as well. Oh, and this is transepithelial transport of organics. And as you can see, this is what we've already talked about, a co-transporter of a solute using the gradient, the sodium gradient. And then on the other side, maybe facilitated diffusion because the concentration in the blood is often not as high as inside the cell. And it says here, of most organic solutes. You have to do that. Don't think in absolutes because there are exceptions to almost everything. So this is figure 4.23. If you want to look it up in your book. Okay. Take a deep breath. Let it out. Take another deep breath. And let it out. Why am I doing this? Anybody know? I could almost cut the tension in the room with a knife. What are you guys uptight about? Oh, I know. You think that these papers up here are a quiz, right? No, it's just it's syllabi. So just relax. And one way you can relax is by deep breathing. Just breathe in, hold it, and then let it out. And this is a deadly time for any kind of lecture course with a darkened auditorium. 2 to 4, it's siesta time, folks. I'm sorry, it's really inhumane. This course used to be 1 to 3, and they changed it for 2 to 4 a couple years ago. I don't know why. It just was something with the summer session schedule. So just relax. What we're going to do is talk about receptors and ligands. You guys want to go pee and then come back at 3 o'clock? Why don't you go get a drink and go out in the sunlight and come back at 3 o'clock? It's a natural break place. So come back at 3 o'clock and we'll start talking about receptors.
How many people are interested in me holding office hours on, actually tomorrow? How many people, honestly, who would come? You're not going to hurt my feelings because I know there's not a lot of lecture material quite yet. Like, who would actually come tomorrow?
You got 2 intrepid students who are scholars. 3 scholars.
Okay, I'm just going to hold them tomorrow. And
You don't have to be there the whole time. Just show up and...
Yeah, whatever question. If you show up for 10 minutes because you have one question, it's more than fine with me. I know how it is. You know, you're not going to hurt my feelings, so I'm here for you, not the other way around. In the future, next week, I will hold them Tuesday and Thursday from 4 to 10, 4 to 6, or from this time, 4 to 10 to 6:20. At Yali's Cafe on Oxford Street. It's above University, but it's below Hurst Street. Everybody know where that is, or?
What's another landmark? Where's the BART station?
Across the street from
Everybody knows where Hurst Street is and University Street is.
It's on Oxford.
It's between those two on Oxford.
Really? I didn't know there was a restaurant over there.
It's on the corner of Berkeley Way.
Yeah, corner of Berkeley Way.
Across from the park.
Yali's Cafe. Never been there. I won't drop in on you because I know this is your personal time with Michael. Okay. Oh, Mike, if you could press the 3rd button down from the green light, that would be great. Thank you. Okay, now we're going to talk about receptor ligand interactions. And this is 3.26. So we're going to a chapter backwards from where we've been. And what we're reviewing here are words and activities. Actually, you know what? Before we go to that one, let me write down a definition for you. Ligand. Now, yes, sir.
I'll turn on the mic.
Oh! I forgot I turned it off. Sorry. How's that? Okay. Some people call this a ligand. A few people could say ligand. So you might hear a lecture where somebody calls it ligand. That sounds very unnatural to me. So it was ligand when I was a graduate student. So it's a ligand. And what that is a chemical or molecule. So let's just say molecule that binds to a receptor's binding site. Now, it can be smaller than a molecule. So here are examples of ligands. I'm going to give you one example is ions, like sodium, magnesium. Oh, well, let's leave it at that. Oh, calcium. Calcium is a good one. We'll talk more about that one. We'll talk about Cal-Gilbert's modulin. Okay, 2, simple molecules, small, simple molecules like NO, which, by the way, was the molecule of the year on the cover of Science Journal. Gosh, when was that? 94? Can't remember. Some hormones and growth factors and neurotransmitters are small or simple molecules. Transmitters. And then 3 could actually be more complex molecules. And complex molecules can be things like steroids and fatty acids, glycoproteins, even sugars, oligosaccharides. I'm not going to write it all out because I ran out of room. What does oligo mean? A few. Okay, so it just means a few sugars that are joined together in a molecule. And when I say the word receptor, that's a generic word, are actually proteins that change when bound by ligand, usually a specific ligand. Receptors can be integral membrane proteins like we talked about yesterday. They can be proteins that are intracellular or even in the nucleus. They can be binding proteins floating about in the extracellular fluid. Enzymes, these all are sort of included in the word receptor. And receptors have, I'm going to just sort of go 1, 2, 3. Oh, this is the pen that when it talks. Anybody want a felt tip pen that's losing its oomph? Recycle. Okay, the binding site is often formed by tertiary protein structure, and I'll show you a slide to show you about that. That is specific for a particular ligand. Specific. a ligand or ligands, usually for a class of ligands. And now I'm going to start showing you slides. I think that's easier. I just wanted to give you some background, so when I use these words, you will know what I'm talking about. Back to this one. This is 326. In this case, we have the protein with its binding site and the ligand, and they're showing in this simple diagram that not only do we have physical shape binding, part of the binding, but we also have chemical nature of the binding site in the ligand that ensure the specificity of binding. So this particular protein, the ligand, or whatever it is folded in such a way that it has a plus, and a negative, and this has a negative, and a plus, because as you know from second grade science class, opposites attract. So that's how we get the specificity. And this is what I meant by the folding of the protein. This is 327 in your books, and this shows the same. It's the same picture, only we have sort of a closer look at what's going on in that receptor protein, and you can see that we've got amino acids on a chain, and they fold into a tertiary structure. This is the primary structure of the same protein. And that was 327. So it shows you how that works. Binding sites have 4 characteristics, and one is specificity. I'm actually going to write these down. Binding site characteristics. 1, specificity. And this is 3.28, showing 3 different ligands and how all 3 ligands can bind to protein X. So protein X has a lower specificity. It's not as stringent. It will bind to anything that has a shape similar to its binding site, whereas protein Y is a lot more particular about what binds to it. It has a higher specificity. In this case, A and B won't bind, but C will. So this is a diagram you might want to spend a little time with, the specificity. Binding sites also have something called affinity. So 2, affinity. So here we have 3 binding sites that are specific for a particular ligand, and one site is a high affinity site. Another site is intermediate in its attraction to the ligand, and then we have one that will allow binding of the ligand, but it is not really highly attractive to the ligand. So that's a low affinity binding site. So this has to do with affinity. Same chemical specificity, but varying degrees of affinity. Another feature of binding sites is something called saturability. So they are saturable. And what that means is that they can get filled up. So this is 3.30 from your book. And this shows with this very low concentration of ligand, you have in this diagram 25% binding, and you can see this is A, so this is where this box corresponds to. The next bump, B, you have a higher concentration of ligand, and you have a higher degree of binding of the binding site. In C, you have higher still concentration, and you have 75% binding. So that's what this dot then refers to, this C. At D, you have such a high concentration of ligand, that you have 100% binding. Actually, in real life, in physiology, you hardly ever have 100% binding. But in this example, we have 100% binding. And as you can see, it doesn't matter when we increase the concentration of the ligand, the saturation doesn't change, because once it's full, it's full. It's sort of like going to a class that has 50 seats in it, and 50 people come in and all the seats are filled, and more people come in, it doesn't make the seats any more full. Well, unless you sit two to a seat, but I don't think these chairs allow that. So that's what's going on here. So this is a saturability of a binding site. And then there's something else, a fourth feature of binding sites. I should write up here, binding sites are, have specificity, affinity, saturability, and they can be competed for. So competition is a feature, and this is 3.31, showing that as the ligand concentration increases, you have different rates of bound or saturation of the binding sites depending on which protein you're looking at. So if you're looking at the blue protein, protein Y, or ligand, oh, I see what they're doing. Okay, it also works if you're using a different ligand for one protein. In this case, they're using the same ligand for two different proteins, and you can see they have different saturation rates because they can compete. They will compete for each other. And in competition, you have something called an agonist. An agonist is a molecule that activates the receptor or the binding site, activates the protein, activates protein. And you have something called an antagonist that does not activate the protein. And I'm going to give you a real-life example. Estrogen has an estrogen receptor. And when estrogen binds to the estrogen receptor, it activates it. There is a chemical, a drug called tamoxifen, that also binds to the estrogen receptor, and it actually has a higher affinity than estrogen. So if you give tamoxifen to a patient, then estrogen will be less likely to bind receptors and activate those receptors because the binding sites will be highly occupied by this drug tamoxifen. Does anybody know what tamoxifen is used for? What therapy? Breast cancer. So if you have an estrogen responsive breast tumor, then tamoxifen will actually block the estrogen's effect and prevent the tumor from growing as much. Tamoxifen has been found to have a very slight agonist quality on the receptor, but it's much, much less than the native hormone estrogen. So what we're doing, we're actually preventing the hormone from binding to the receptor through the process of competition. So two kinds of competition, or two kinds of antagonists. Antagonists, I should write this again. Antagonists can be competitive, like tamoxifen, or they can be non-competitive, which means that instead of binding the binding site, they bind something else on the receptor, which changes the shape of the receptor. So competitive binds to the binding site. And non-competitive binds elsewhere. And this will become more apparent what I'm talking about when we go through the rest of this set of slides.
What?
Getting the light comes on. Okay, this is 3.32 in your book, and it talks about, well, it's an introduction to binding site regulation. And one way to regulate a binding site is to change the shape of the protein. And this shows what I was just talking about, the allosteric binding of the protein somewhere other than the functional site will actually change the activity of the functional site. I'm going to come back to this one. So I will put this one by, and I want to tell you about something else. Okay. So what I've talked about is I've reviewed ligand and receptor and binding site. 2 was characteristics of binding sites that included specificity, affinity, blah, blah, blah. 3, binding site regulation is where we are now. And one way to regulate the binding site is to change the rate of synthesis or degradation of the protein. So if you want a cell, this is an L. If you want a cell to be more responsive to a ligand, what do you do to the rate of the protein synthesis? You increase it, right. Another way is through changing the shape of the protein itself. You all know what delta stands for, right? Change? Okay, good. And then there are some non-specific mechanisms that can change the shape of the protein. Specific. And those actually affect all receptors of that type. Non-specific will affect all receptors of that particular type. And those factors could be pH, temperature, presence or concentration of certain ions. So any of you who've done enzymatic reactions know that if you add a little magnesium to a certain enzyme mixture, that you'll get a better result than if you don't. So the ions actually help. Okay. Allosteric modulation is specific. And what happens with allosteric modulation is the regulatory site, a regulatory site, somewhere else on the molecule, binds a modulator molecule, changing the protein shape. And then, when the protein changes shape, the functional site changes. And it either becomes activated or inactivated depending on what system you're looking at. So that's allosteric modulation. An example of allosteric modulation. Notice the colors. Calmodulin has actually 4 binding sites for calcium. And it's usually bound in most cells on 3 of those sites. But in order to bind that 4th site, you need to increase the calcium concentration greatly. And that is how you get the calmodulin to do its thing. It has to have all 4 of the calcium binding sites bound. And when you bind the calcium, this last calcium binding site with a calcium ion, so that would be calcium ion, it actually changes its interaction with another protein, and the protein itself then can have an active site or a functional site that can now be useful in some kind of activity. It's usually an enzyme. Calmodulin is usually connected to an enzyme. Okay, the other kind of, where was I? This should be C. C, covalent modulation of the protein. It's usually phosphorylation is the nature of the covalent modulation. So back to this slide. And this is 3.32. And this shows you that when you have allosteric modulation, a modulator molecule binds to some site, not the functional site, changing the shape of the protein and activating the site or deactivating it, depending on what the modulator's purpose is. Or you can have binding of the protein at a phosphorylation site, which then changes that active site in the same way, or the functional site in the same way. So covalent binding of the protein of adducts, usually phosphorylation, also can modify. So these are 2 different ways to modify a protein, a receptor protein. What's the source of the phosphate in phosphorylation? ATP, so it takes energy, doesn't it? Okay. So sometimes you will have receptor molecules that are regulated by more than one process. So this is sort of a generic enzyme from your book, 3.37, and the active site is modified by binding of a covalent binding site or binding of allosteric sites, or it could actually be inhibited by similar sites. So you can either activate or inhibit, it depends on the receptor protein. Examples of multiple site regulation of a protein. This is such an old slide. I get really nostalgic when I look at it. But you have one very similar to this one. This is showing primary active transport, and here is an allosteric modulator that then, or allosteric modulation of a transporter, first by the binding gesundheit of the solute that is about to be transported, and also covalent modulation by phosphorylation. So we're actually changing this molecule with 2 pieces of information. One is the binding of the transported solute, and one is phosphorylation. So that's one example of a type of protein that is modulated in two different ways. Another is the ubiquitous sodium-potassium pump, and this is an older slide of the one that you have in your book that shows we have covalent binding of phosphate plus the binding of the solutes to be transported together, change the conformation of the protein. Factors that affect the rate of enzyme-mediated reactions. This is, I think, 338 in your book. And you can see that the concentration of the enzyme, the concentration of the substrate influence the rate of the reaction. Also, the activity of the enzyme as affected by the things we just talked about, allosteric and covalent activation or inhibition. This is not in your book. This is from Campbell's Biology. I showed this to you because I think it's a wonderful picture. This shows feedback mechanism. Feedback inhibition in a process of going from a substrate to a product. So we're changing threonine into isoleucine. And that is a 5-enzyme set of steps. And you can see that as you go through this, you use a different enzyme. So enzyme 1, enzyme 2, enzyme 3. Once we get to the end product, if the end product is getting too concentrated to prevent buildup of this end product, as the concentration increases, it will actually inhibit the first enzyme and put a stop to this whole thing until the concentration of isoleucine goes down enough so that the inhibition doesn't happen. Tell me, why do you think it intervenes all the way up here on enzyme 1? Why can't it just feed back on enzyme 4 or enzyme 5? What's the point? Save energy, yes. It takes too much energy to make all these enzymes and to go through the process of getting to these intermediates. So it stops it cold at the very beginning, and then you don't waste a lot of ATP making those enzymes. Or raw materials, because each enzyme is made up of amino acids. Okay. I'm going to recap what we just talked about. You guys okay? Yeah. Okay. If you have questions, be sure and holler or raise your hand. Okay, so first of all, the ligand, remember the ligand, binds the functional site. Oops, ran out of room. Hate it when that happens. Of the receptor protein, which is mainly built on the tertiary structure of the protein. We'll talk more about that as we get into endocrinology. It has to do with the shape of the folding, where the hydrophobic regions are in the protein, and so forth. And those also influence specificity. So the ligand binds functional site specifically, it's hard to talk and write at the same time, and and with affinity. Something else you need to remember, receptors are saturable. I'm really digging into this, folks, because I don't want this to be a misconception 3 weeks in. This is all basic stuff some of you have already had, but it's important that everybody be up to speed on this. Ligands can compete for a receptor. Affinity can be modulated through two different ways, allosteric, and I gave you a couple of examples like Cal-Gobert's modulin and covalent modulation. Covalent modulation is usually done through phosphorylation. This is a biggie, phosphorylation.
Question.
Yes, sir.
Are there any cases where it's not phosphorylation?
Not that you need to worry about. I can't think of any off the top of my head. Yes.
And either of them can inhibit or
Right. Remember that one slide that showed all those different sites? You could have a covalent binding site that inhibits, or you could have a covalent binding site that actually activates the protein. So I want to give you another word, and that is the word kinase. This word will crop up over and over again. What does a kinase do in general? It catalyzes, it's an enzyme, isn't it? It catalyzes phosphorylation. And usually what it does is it catalyzes phosphorylation of a specific protein. So you will have all kinds of proteins that have their own personal kinases. So keep this word in your head, kinase. Yes?
So all those things we were talking about earlier with like the transporters and all that, when things bounded with those were all ligand.
Most mediated transporters are either covalently modified by phosphorylation, or else, in the case of facilitated diffusion, are allosterically modified by not a ligand, but by the solute that's being transported.
So that solute is not called a ligand.
Not in that case, no. Because it's not binding and then coming back out. It's binding and being shuttled through the membrane. It's not going to be an issue for you in this class, okay? But if you want to come and we'll chat about it, and then we'll make the distinction clearer. Okay. Stand up. It's 3:30, folks. It's siesta time. Stand up and stretch. If you're next to somebody you know and really like, maybe do a mutual back rub. You know, just
Because I mean, ligands are binding.
yeah. The way it's used in physiology is ligand usually refers to something like a growth factor, an ion that is activating the protein, or an agonist.
Not so much like involved in transport.
Not so much the transport.
Can you say that ligand is involved in like involved with that enzyme? When you're associating with an enzyme, you call it attached to a ligand?
Same can be said true of an enzyme or receptor. Not so much a transporter, maybe a channel. Channels are often ligand gated. So the word is generic.
The way we learned in biology was the only time we ever used the word ligand was for enzymes. But it makes sense. It's just
What, when it was an enzyme?
enzyme and ligand.
We're going to bump it up another level. Okay, you can sit down now. I just wanted you to get up and get the blood out of your butts and into your brains. And if at any time during the class you can feel yourself going, what'd she say? And I'll give you, there's almost always somebody back in the cheap seats who's going semi-recumbent. And as soon as you start to go horizontal, you know what your body says? Nap time. And look at the time. It is nap time. So don't lie down in class. Sit up. And if you can feel your brain freezing, or you feel yourself getting really tired and sleepy, stand up. Maybe I've seen a lot of people who have back trouble will stand up during class and they'll just listen this way, just to get their blood moving and to get their back straight. And some people can't sit for 2 hours. You notice I don't sit for 2 hours. I stand for 2 hours in the afternoon. So, and an hour and a half in the morning. So, do feel free to move around the room if you want to. Okay. There is a word I wish you to learn. We're moving into a new realm now, and the word is homeostasis. Some people say homeostasis. I might say it either way. Who knows? Homeostasis. What does homeo mean? Same, right? So from Greek it means similar or same. Stasis, I'm going to tell you a little story. I love stories. When I was in Greece, not last summer, but the summer before, I saw all the stop signs. This should be in red. And the stop signs said this. Okay, Greek scholars, what does that mean? Stasi. Stasi. Which means stop. So, homeostasis doesn't mean same stop. What it means is stay the same or don't change. Keep it kind of within parameters. So during the next 8 weeks, I will be asking you often, how does this system or organ contribute to the maintenance of homeostasis? Or maybe even you will be asking each other and yourselves, how does this organ or physiological process contribute to homeostasis? This is big. It's huge. It's more important than getting a mate. Trust me on this. It's more important than winning the battle. Homeostasis is about keeping things real and even in the body. So always ask yourself, what does this process do to contribute to homeostasis? Also, while you're asking yourself questions, ask yourself, what is the evolutionary advantage to having this system or process in place? So the two things you need to keep in mind, and one is homeostasis, and the other is the selective or evolutionary value of a process. And when I say the selective value, I mean, how come we, after millennia of natural selection, are the way we are? Why do we have this complicated 5-enzyme pathway to get from threonine to isoleucine? What is the point of that? Well, sometimes the point is, was that a computer going on or a cell phone? Okay. So sometimes what happens is one little piece of a pathway may evolve and confer some kind of advantage to the organism. Then another little piece of a pathway over here may evolve or be selected for, and then there may be another aspect that actually joins those two pathways, and that's why we have these lengthy and complicated pathways, because it's not intelligently designed, folks. Don't let anybody tell you that the body is too complex to have been the product of natural selection. It has to be intelligently designed. I'm telling you, intelligent designers would have made it a lot simpler. I'm telling you, an engineer would not have had us be so complex. So ask yourself these two questions: selective value of a process, and how does that process contribute to homeostasis? These are the kinds of questions I just adore. Hint, hint. I'm giving you a lot of help. The internal environment is relatively stable. So for instance, the kidneys work really hard to keep your plasma, your blood plasma, at 300 milliosmoles. And the reason for that is they want everything to be isotonic to the insides of cells. This is a good thing. So let's say you go to Jupiter Cafe and hoist a few. Okay, your kidneys are going to do what? They're going to increase the removal of water because you have all this fluid coming into you. Because it doesn't just go into your bloodstream purely as beer, okay? It comes in as water and alcohol and carbohydrates and all those good things. But what will happen is you have all this water, and that's one of the reasons, not the only reason, but one of the reasons that you have to go to the ladies' or men's room when you are in the middle of a beer bash. Or if you go backpacking in the Sierras, where you don't have a keg of beer. In fact, you have a little bitty canteen full of water. And you drink it sparingly. You can't drink from the stream because the stream has giardia in it. So you're not drinking as much water as you should be, and you're sweating a lot. And that means that the concentration of your plasma is getting greater because you have less water. What will your kidneys do then? They'll slow down. You basically have to get rid of about a half a liter of water a day just to get rid of all the nitrogenous waste and other stuff you have to do. But you can, if you're at Jupiter, you could be getting rid of liters worth of water, depending on how much you're drinking. But that's because the homeostatic mechanisms of the body are kind of like a thermostat. They try to keep everything within balance. In fact, everybody put your hands like this. Don't be shy. Everybody's doing it. No, you don't look silly. I'm the only one that looks silly, okay? And have your right hand go down, and then your left hand, and then your right hand goes up. So this is homeostasis, folks. Okay. So, yes, sir. Where's Jupiter? Well, it depends on how you enter it. You can get it. The main door is on Shattuck, but you can get to it from Oxford. Great pizza. Is the beer good too? Okay, I wouldn't know. All right, so this is a diagram. I think this is 7, 8 in your book. The new set of overheads didn't seem to have one. And it's just a balance diagram showing what's going on in the body. So you have food coming in through the GI tract and air coming in through the lungs, and then you have synthesis of materials in the body, and that's adding stuff. And then you have net loss. You metabolize the food, you metabolize all kinds of things. You will break down things that you made, like hormones. You will have excretion from the kidneys and the lungs of materials, and also from the GI tract and the skin when you sweat. So what happens is, in the middle here, you have places where these things are stored or not. Storage deposits like fat, for instance, or iron in the liver, in the blood. And what happens is you are bringing new stuff in, you're getting rid of old stuff, but you have to keep it kind of in a balance. See what I'm doing? You have to keep it in a balance, because if you get too far in deficit, then you will be encouraged by your body to get more of whatever you are deficient in. Like, for instance, if you're dehydrated, you get thirsty. If you get too much of something, then you will be encouraged to get rid of it. And that's to maintain homeostasis. So outgo needs to equal income. And if you are bringing in more calories than you're getting rid of, it gets stored in adipose. Well, we'll talk about that later when we talk about metabolism. This is an example. Oh, wow, that was a good sneeze. Good sneeze. Good sometimes. Okay. Yeah, don't try to hold in a sneeze. I used to work with a woman, and she sneezed like this. She sneezed about 15 times in a row. I say, if you just sneeze once big. And there's a reason for a sneeze. Very important. And we'll talk about that when we talk about the respiratory system. Okay, so 1.3 in your book gives you an example of one homeostatic mechanism. There are many homeostatic mechanisms. This is just one of them. Let's say that the temperature goes down. Not in this room. It's almost always hot in here, isn't it? Or are you guys cold? It's because I'm standing up in front of the hot lights. Okay, so the room temperature goes down, and you start to lose heat from the body. So your core temperature will go down. And the way you respond to a drop of your core temperature is a constriction of skin blood vessels, because the capillaries in your skin actually radiate heat. So when you're really hot, you know how you feel hot all over and your skin kind of gets pinkish and flushed? That's because your capillaries are dilating so you can give off more heat. When you're cold, then your blood vessels in the skin constrict. I had a friend, we were swimming in the ocean off Carpinteria when I was, 15, and she was very blonde and blue-eyed, very pale, and her skin was turning blue, and her lips were actually turning kind of a purplish blue, and that's because she didn't have a lot of blood coming to her skin, because they were all constricting, because that ocean is cold. It's fed by the Alaska current. Another thing you do is you have an inclination, a behavioral inclination, to curl up in a ball. So let's say you go to sleep at night and it gets really cold overnight. How do you wake up? You wake up like this. You wake up like a cat. You're sort of like, and that's to conserve heat. Actually, if you're overheated, you will wake up like this. Another thing you do is your muscles are stimulated to contract, not to move you or change your position, but to cause you to shiver. And what the shivering does is it increases the waste heat from muscle contraction, increases your core temperature. Another thing that's not on here is smooth muscles in your skin will contract to raise your hairs, and that will cause goosebumps. The goosebumps don't keep you warm, but maybe if you're hairy, it will keep you warm. So, and you know, we evolved from hairier beasties than we are, and that's one way we keep warm. So when you feel the hair going up on your arm, it's just because you're cold, you need to keep warm, or maybe you're scared. That's another thing we'll talk about. Okay, so this is just one homeostatic mechanism to keep all of your levels at a set point, to keep your temperature within certain parameters. Your temperature should be around 37 degrees centigrade. And it could be maybe 36.5 degrees centigrade. It could be maybe 37.8 degrees centigrade. But it should not be 32 or 30 degrees centigrade. That's very bad. It certainly should not be 45 degrees centigrade. At that point, your enzymes are starting to cook. So you really want to keep that body temperature within the set point. I talk too much. I should let you guys talk. Who wants to come up and teach this? Okay, so this is a generic picture showing what I was talking. Remember, I was talking about feedback inhibition in that enzymatic pathway. This is called negative feedback. So we have a stimulus. This is like a little flow chart. And in the old days when we did BASIC for computers, we used these. Do you use these nowadays in computer programming? Probably not. Okay, so stimulus is transduced by a receptor, and then the receptor sends the message up an afferent fiber to the integrating center, which is in the central nervous system. The central nervous system processes this information, sends out information to an effector, which then causes a response, like shivering, for instance. And then when you shiver, it heats up your body. And what happens is the stimulus, in this case being chilled, will then not be such a big stimulus. Your receptor will say, "Oop, not so cold anymore." The integrating center will say, "Well, then slow down your shivering." So you will actually see that your central nervous system talks to the rest of your body in order to make sure that everything is kept in balance. This is something that's not in your book. Well, this picture is not in your book. It used to be. This is an old Vander slide. So this is we were talking A, about a steady state, homeostatic steady state. And the first thing I told you about was negative feedback. That last slide. Okay, this is error signal. Let's use our being cold example. Let's say it gets cold. This is too bad because this is going up. Well, I have another one for that. Let's just say we have an environmental change that causes changes in a given body point or value, and you will eventually have a change to a new steady state as long as this environmental change is in place. The difference between your set point, your regular steady state, and this new steady state is called error signal. Can anybody tell me why? That's another favorite question of mine. Why? Why do we have error signal? Why doesn't the homeostatic mechanism just bring you back down to your steady state? That's a good answer. However, that's not the reason for error signal. Say again? It will take energy probably, but that's not the reason. Why don't we just go back? Yes? That's it. If we went back to our steady state, the homeostatic mechanism would be turned off. And then we'd change again, and then it'd have to be turned on again. Then we'd go back to steady state, and it'd be turned off. And then as long as this environmental perturbation persists, we will have to keep turning it on and off and on and off. But instead of doing that, as long as the environmental perturbation exists, we will have a new steady state that exists as long as this environmental change is in place. And that keeps the homeostatic mechanism working throughout the whole thing. So if I were to be out jogging, which I just don't do anymore, but okay, let's say I'm jogging and I get really hot inside and I start to sweat. And the sweating actually does evaporative cooling. It cools me off. And maybe I'll even take off my sweater or my jacket so that I'll have more ability to get rid of heat. And my vessels will dilate in the skin so I can give off more heat so that the core temperature can go back to set point. But it never goes back to set point as long as I'm jogging. Because as long as I'm jogging, there will be error signal to keep all of these homeostatic mechanisms working. In fact, sometimes after you finish jogging, you notice how you still continue to sweat and have a flush and not want to put your coat back on because your core temperature is still a little bit hotter than normal. And that's just to keep those homeostatic mechanisms at work, keeping you from overheating. So that's the purpose of error signal. Error signal is in order to maintain the homeostatic response to an environmental variation. If you return to a normal set point while the change persists, you just have to keep turning it on and off and on and off. So error signal is the difference between your usual steady state set point and your new steady state that exists as long as the environment has changed. Yeah.
I'm having a hard time like physically imagining what an air signal is. Like I know that negative feedback when you have a thermometer when you're trying to act or something. So like what would the air signal be?
It would be whatever change the environmental perturbation causes. For instance, cold or heat. Let's go back to Jupiter. Let's say you eat a big bowl full of salty peanuts. So while you're eating those peanuts, your salt concentration in your blood is going up. And during that time, your kidneys are going to have to change the speed with which they excrete salt to preserve your osmolarity. And as long as you are eating those peanuts, your plasma salt level will not be set point, it'll be a little bit higher. And the difference between where it is while you're eating the peanuts and the steady state set point is your error signal. We will revisit this tomorrow. Okay, go home. Or go to the pub.
Y'all